Now let's divert our attention to the mitochondria, where we'll find our electron transport chain. There are quite a few fancy figures out there, but I'm here to make this as simple as possible, because knowing all the minute details is not going to be important for the boards. However, the things that I'll be talking about will be. So we have five major complexes, named complexes 1, 2, 3, 4, and 5. Each one has a very unique function. The blue line is going to represent our inner membrane of the mitochondria, and our mitochondrial matrix is going to be on this side, while our intermembrane space is going to be up on this side. So this is kind of an orientation. This is the middle of the cell. This is more towards the outer membrane of the mitochondria. So we have pre-made activated carriers like NADH and FADH2. These are going to dump their electrons at these complexes and ultimately make some ATP. And where did we make these carriers? Well, we made some pre-made NADHs in glycolysis using the enzyme pyruvate dehydrogenase and also in the TCA cycle at various steps. We also made some FADH2 molecules in the TCA cycle. Our NADHs are going to enter at complex 1 while our FADH2s enter at complex 2, which happens to be the enzyme succinate dehydrogenase, which we found in the TCA cycle. As electrons pass from complex to complex, they're going to create a proton gradient, which is going to be used to drive the production of ATP. So as it passes through complexes 1, 3, and 4, we're going to create a proton gradient. So eventually we're going to have a whole bunch of hydrogen ions, for example, on the inner membrane space. So we have a lot of hydrogens over here, but not over here. Our fourth complex, which is known as cytochrome C oxidase, is going to transfer electrons to the final electron acceptor. Do you remember what that final electron acceptor is for the electron transport chain? Well, it's oxygen, which is ultimately going to be used to create water. So as electrons get transferred from complex to complex, final complex is going to be complex 4, which is going to then dump its electrons on oxygen, which combines to form water. Now, without oxygen, there is no final electron acceptor. So the electron transport chain cannot function if you don't have oxygen in the body, and thus you must rely on anaerobic metabolism if oxygen is not present. Complex 5 is going to be called ATP synthase. So it has a unique name, and it is going to be used to create ATP, as the name implies. It's going to use this hydrogen gradient, which we formed by using complexes 1, 3, and 4. So it's going to drive this hydrogen gradient to form 3 molecules of ATP for every NADH molecule, and 2 molecules of ATP for every FADH2 molecule. Since FADH2 starts at a later complex in the pathway, it's not able to pump as many electrons out. Notice how an NADH is going to be able to pump out 1, 2, and 3 different spots in our system, while FADH2 is only going to be able to pump out 1 and 2 at 2 different complexes in our system. So we're going to have less hydrogen gradient performed when we enter in an FADH2 compared to an NADH. Now, complex 5 is going to take an ADP and combine it with a free phosphate to form an ATP molecule. So this is going to be our high energy ATP molecule. So a lot of stuff going on, but we're going to clear that off now. We can now step aside and introduce 3 scenarios where oxidative phosphorylation becomes poisoned or inhibited. The electron transport chain inhibitors, such as rotenone, cyanide, antimycin A, and carbon monoxide, are all going to directly inhibit the movement of electrons from complex to complex. This is going to prevent the formation of this nice proton gradient, which we drew up here, and therefore we're not going to get the synthesis of ATP. Rotenone is going to inhibit complex 1, antimycin A is going to inhibit complex 3, and both cyanide and carbon monoxide are going to inhibit complex 4. Oligomycin is an ATP synthase inhibitor. What does that mean? Well, it's going to directly inhibit complex 5, our ATP synthase. This allows for this proton gradient to be backlogged. We're going to create a logjam in the electron transport chain. these protons are not going to go through complex 5 to create our ATP, so we get a buildup of our protons, thus we get a backlog of everything else, so we're going to increase NADH levels and increase FADH2 levels because we're not able to properly run our cycle. And then lastly, how do uncoupling agents work? Well, our uncoupling agents such as 2,4-dinitrophenol, which was used as a weight loss agent, aspirin, and also thermogenin, which we'll find in brown fat, these three are going to increase the permeability of the inner membrane. So remember our double blue line here is our inner membrane. So this is going to allow protons to sneak through this inner membrane without actually going through complex 5 to create our ATP. We're increasing the permeability of this membrane. This results in an increase in oxygen consumption, since an increase in electron transport chain attempts to compensate for this permeability, this loss of our hydrogen-proton gradient. Still, ATP cannot be created without this nice proton gradient, and the increased electron transport activity is instead dissipated as heat. We're not creating ATP, we're creating heat instead. This is why an aspirin overdose can cause fevers. Then also, this is why babies and hibernating bears can stay warm, since they have lots of brown fat, and this inner membrane permeability is going to create heat. So let's test our knowledge. A 4-year-old girl swallows some cyanide-containing rat poison from underneath the sink. How does this affect her ATP production? Remember that cyanide is going to directly inhibit the electron transport chain, specifically complex 4. So this is going to decrease the proton gradient and block our ATP synthesis. Those electrons are not able to transfer efficiently from complex to complex, specifically because we blocked complex 4. Therefore, the body isn't able to create ATP via oxidative phosphorylation. We're going to now shift gears and talk about gluconeogenesis. What does the name gluconeogenesis mean? Well, it simply means making new glucose molecules. So gluconeogenesis, shown here, is basically the glycolysis pathway, but this time in reverse. It's going to occur primarily in the liver, because when the body's low on blood sugar, the liver is going to use gluconeogenesis to pump out more usable glucose for the body. This way, the body remains in a euglycemic state, even during fasting. There are also enzymes found in the kidneys and the interstitial epithelium. This pathway is going to begin with a pyruvate, and we're going to end up with a glucose. And this is going to pretty much use the same enzymes that we saw in the glycolysis pathway. However, there are going to be 3 irreversible steps that we talked about in glycolysis. The body is going to use 4 unique enzymes to bypass these 3 irreversible steps of glycolysis in gluconeogenesis. Now, before we talk about these 4 enzymes, do you remember what cofactor is required for the carboxylase enzymes? Well, hopefully you're thinking biotin. When you hear carboxylase, always think biotin. The first key enzyme of gluconeogenesis is pyruvate carboxylase, which is going to convert a pyruvate into an oxaloacetate. This occurs in the mitochondria, and it requires biotin and ATP. This step is activated when there is a high concentration of acetyl-CoA, since this means you don't need to be producing more acetyl-CoA and ATP, and thus you'd be better off storing energy as glucose in the form of glycogen. Next, an enzyme called PEP carboxykinase is going to convert an oxaloacetate into a phosphoenol pyruvate, or PEP, in the cytosol. This is going to use GTP as a source for that phosphate group. Fructose 1,6-bisphosphatase is the third irreversible enzyme, and this is going to convert a fructose 1,6-bisphosphate into our fructose 6-phosphate. And finally, glucose 6-phosphatase is going to remove phosphates from our glucose 6-phosphate to form glucose, which is then going to be free and released into the bloodstream to be used by the body. This last step occurs primarily in the hepatic endoplasmic reticulum, and it does not occur in muscle, since muscles actually lack this enzyme. Glucose 6-phosphatase is also the enzyme that is mutated in von Guericke's disease, which we'll talk about soon. So, in summary, gluconeogenesis occurs primarily in the liver, and it creates glucose from glycolysis, fatty acid, or TCA cycle intermediates. It should come as no surprise that a deficiency in any one of these four gluconeogenic enzymes can cause hypoglycemia, or low blood sugar. On test day, how in the world can you remember the irreversible enzymes of gluconeogenesis? Just remember that the gluconeogenesis pathway produces fresh glucose, where the first P is going to stand for pyruvate carboxylase, the second P is going to stand for phosphoenol pyruvate carboxykinase, the F is going to stand for fructose 1,6-bisphosphatase, and the G is going to stand for glucose 6-phosphatase. Now, how else can we make glucose molecules? Well, here we see our TCA cycle, which we already touched on. Now, the odd chain fatty acids can serve as glucose sources, since they yield propionyl-CoA, which can then enter the TCA cycle as a succinyl-CoA. Even chain fatty acids, however, cannot be used as a glucose source, since they can be only broken down into acetyl-CoA, which then disappear as CO2 by the time it reaches oxaloacetate. So we're losing CO2 molecules, we're losing those carbon groups. Let's quickly cover the HMP shunt, also known as the pentose monophosphate pathway, which we can kind of see here. This is going to occur in the cytoplasm of a cell. I'm only going to touch upon the important stuff for the USMLE. There's a lot of enzymes and intermediates that I'll be neglecting. However, I'm here to get you maximum points on test day, so really focus on the points that I'm going to be talking about. The purpose of the HMP shunt is to create molecules of NADPH and also ribose sugars. This occurs primarily when the glucose 6-phosphate is abundantly available. The HMP shunt can be broken down into two different steps. On the first hand, we can see the irreversible oxidative phase. Then on the other hand, we'll be talking about the reversible non-oxidative phase. So in the irreversible oxidative phase, the part that we'll be talking about first, the first molecule of NADPH is produced by the enzyme G6PD, or glucose 6-phosphate dehydrogenase. Then another molecule of NADPH will be produced later on. Do you remember what NADPHs are used for within the body? NADPHs can be used in reductive and anabolic reactions, such as in glutathione reduction in red blood cells and cholesterol synthesis in the liver, which is why this pathway is most active in lactating mammary glands, the liver, the adrenal cortex, and red blood cells, or erythrocytes. If the cell already has plenty of NADPH available, this pathway will become shut down since NADPH is going to inhibit the rate-limiting step in this reaction, G6PD. So it's going to actually inhibit it. The pathway also consists of a non-oxidative reversible phase. So this right here, what we're looking at, is the irreversible oxidative phase. Now let's move on to the reversible non-oxidative phase. So the non-oxidative reversible phase will create ribose 5-phosphate derivatives using a phosphopentose isomerase and transketolases. These ribose 5 derivatives will be used elsewhere in the body, such as in nucleic acid synthesis. So these transketolase enzymes are going to require vitamin B1, and thus is often used to test for a vitamin B1 deficiency. Also, lastly, let's notice how the end products of the HMP shunt feed back into our glycolysis pathway. This way, the body can use the HMP shunt to make NADPH or ribose derivatives without creating too much waste, because the end products would feed back into our system. So a 2-year-old girl is born with a glucose 6-phosphate dehydrogenase deficiency. This interferes with which step of the HMP shunt? Well, it'll interfere with the first step, the irreversible oxidative phase, or the first reaction of the HMP shunt, which is going to convert the glucose 6-phosphate, remember from our glycolysis pathway, into a molecule of CO2. It's going to create 2 NADPHs and also that ribulose 2-phosphate molecule, which will then go on to the second part of the HMP shunt, which is the reversible non-oxidative phase. However, the first step is going to be glucose 6 phosphate. We're going to get an NADPH produced.